So we're going to talk about follow-up. So what do you do after you randomize people? What should be in the contents of the follow-up, frequency of follow-up, importance of complete follow-up, and then that kind of leads us into talking about compliance and adherence, how to measure it, how to maximize compliance and adherence. And then I wanted to spend a few minutes talking about the consort guidelines, which are guidelines regarding reporting follow-up and compliance in clinical trials. And then the last couple things, talk about ITT, intention to treat, definition, different types of this. It's kind of a confusing issue. And then if we have a few minutes at the end, there were a couple of data analysis issues from last time that I wanted to revisit or bring up. I didn't have time to talk about. Okay, so follow-up. So we've talked about subject selection and different kinds of things. So follow-up is now what happens farther along in the study after you randomize people. And obviously what you need to do when you're designing a trial is carefully lay out what the procedures are to be followed. I think we'll talk in a later lecture a little bit about efficiencies in clinical trials. And this is one of the areas where you can really gain a lot of efficiencies and save a lot of money by being very parsimonious about how you do measurements. And that also may actually increase adherence, because if the patients feel that the follow-up visits are odious, they're 6 hours long, they may not want to come. So this is something really important to think about. And I think that the questions, which I should have put it at the top of a slide, but they're at the bottom. When you're thinking about measurements in a trial, and I'm going to go through a couple of examples. Do you need to do them at all the visits, or can you do them only at some of the visits? Are there measurements where you don't have to do it on every patient in the trial, like only on a subset of patients? Or maybe there's a measurement and you say, well, everybody's always done labs before in this particular context, but maybe there's really no reason to do safety labs. So, those are all the questions I think you need to ask yourself. And often people will go through those little charts that you make up that talk about measurements and then visits, and then say, do I really need to do this? And really take a red pen to it and see if you can increase the efficiency. So, different, sort of like, that leads to thinking about different kinds of styles of follow-up. So the intensity of follow-up, it depends really on the needs of the study and on the budget. And you think kind of at one extreme, you could think about these sort of maximally intense studies, like an inpatient GCRC type study, where you have people there and you're measuring them constantly, 24 hours a day for, say, 4 days or a week or something. Phase 1 studies, sometimes phase 2 studies, there's often a need to do a lot of measurements because you're very carefully monitoring safety. So that's not unusual. At the other extreme, you might have kind of a minimalist study, and I'll talk about a couple of examples of those, where you only have a few follow-up visits and a few measurements. And the cost, sort of the cost differential, as you might expect, is huge in these things. And there are, of course, advantages and disadvantages to each type. Here's one example, and this is the zoledronic acid trial, the so-called Horizon trial, that we published a year and a half ago, or almost 2 years ago now, in New England Journal. And I put that on the website if any of you have computers and you want to just look at the study. But I'm just going to use it for a couple of examples. This is a study where we had 7,700 participants. The primary goal was to look at zoledronic acid and its effect on vertebral and hip fractures. Secondary outcomes included bone density and markers of bone metabolism. Well, both for bone density, and I'll give you more detail about markers of bone metabolism, there was a question of whether we really needed to measure those in everybody and how often we needed to measure them. And some of the investigators said, oh, it would be really cool to have measurements on everybody at every visit. So to take blood and measure, say, bone markers. But the people cutting the checks, the pharmaceutical company, in this case was a Swiss company, so you can imagine they don't like to spend money. Anyway, they didn't want to spend money, and so what we ended up doing is doing the markers of bone metabolism on a subset of participants. And this is kind of a convenient subset. It wasn't a random subset, and it was about 500 participants, which is only about 7% of the total. Now, another thing, I would say, why convenient, why not random? Well, it turns out that this study was done all over the world, and it was very expensive to ship ship blood, for example, from Asia and from Russia, and where it was much easier to ship blood to a central lab from North America, South America, or Western Europe. And so they took a sample of participants from those sites that were in those places where it was easier to get a hold of them. And we also decided for the bone markers, rather than doing it at all the visits, and sometimes there were visits every 3 months, we did it every 6 months. And then the cost savings compared to doing that measurement on all participants at all visits was over 95%. So if you really put it all together, we saved a huge amount of money and many, many millions of dollars on this particular study by doing it. And these are the results for one of the bone markers, and just not to really show you the results, but just to show you that this was the number of people who had the baseline measurement. You can see the number as we go along. You kind of lost some people along the way. But the point is that we still had enough people to be able to make inference about what the markers did. And you can see the confidence intervals are fairly narrow. So again, just a point if you're designing a trial to think about, can some measurements, generally not primary outcome, but can some measurements be done less frequently? Now, in sort of an extreme case, you can talk about something that's called the large and simple trial. It's been discussed a lot in the literature. And the idea of the large and simple trial is, rather than getting a bunch of people, recruiting them, seeing them every month in the clinic, talking to them on the phone all the time, you get a whole lot of people easily and then randomize them and do as few follow-up measurements as possible. And these kind of trials of course, can be difficult to do in practice. But there are some good examples, and now with the internet, people are talking about the possibility of following up people via websites, for example, so you don't have to see them. But what's classically been done as the most streamlined type of large and simple trials are trials in which you follow people by mail. So, for example, the Physicians' Health Study was a randomized trial where they randomized aspirin or placebo. And not only did they follow them by mail, but they mailed out the drugs. And they followed them by mail. It was a study of physicians who may or may not have been compliant, but probably were fairly compliant. And they used, then to get the endpoint data, they used data collected for other purposes. For example, the mortality data you can get from national databases. And then if you're within a medical system, not for this particular study, but you can use, for example, Medicare data is available under fairly strict confidentiality, but you can do it. If you're in a system like Kaiser is a wonderful system for doing these kind of studies because you collect the follow-up data, the endpoint data, as part of their normal medical care. And then in Europe, for example, where they have socialized medicine, especially in countries like the UK or in Scandinavia, they really know every time anybody has a hangnail, that's on the national database. So it's a really good way to if you can embed a study where you can save a lot of money doing the study. And again, a lot of people are talking, and some studies are doing now, internet follow-up of participants. I know a few years ago, maybe 10 years ago, we were talking about this saying, well, that's crazy. Nobody over 50 would ever have email or ever be on the internet. So it's now become very practical. I'm sure a lot of you, your grandmothers and parents are all very avid internet users. So this is definitely a thing that can be done. Okay, so here's an example of a large and simple trial, and the British are particularly good at doing these. There are a lot of British trials. This is a British trial, 2,700 men and women from a general medical practice, and they were studying vitamin D. So what they did is they distributed oral vitamin D, 100,000 units, which is a lot, but it was given only 4 times per year orally, and they were looking at the prevention of fractures. So vitamin D is a particularly good thing to study, something because they mailed out a pill every 3 months. They got your pill in the mail. Hopefully people would open the mail and take the pill. So that's a nice way to do it. And they sent it all by mail. It was vitamin D or placebo. And they ascertained fractures via post, as they say in England, so by mail. And also in the UK, they have the National Health Service database. So they had 2 ways of ascertaining fractures. And then the mortality was assessed by national statistics. And actually, amazingly enough, they found a decrease in fracture risk. And these are just the results, which are not so important for the discussion as a method.